Okay, good afternoon, everyone. Almost, yes. And I hope you have, this is a problem that I have given you before, and I ask you to solve it. I think some people have attempted and did well, and some of you have not yet finished. I'm still waiting for everybody's answers. So keep that in mind. There will be, throughout this course, we will discuss a lot of problems. It's quite common that very similar problems will be repeating in the exam. Okay, so you better try to do well and try to practice them more. Okay, in the last class we discussed about several of these, the linkers and adapters. We discussed that the linkers are oligonucleotides, which are double-stranded. That means you make one single strand at a time, and then you anneal them, and that's what becomes a linker, double-stranded. And the other part, the point is that the linker has the restriction site, complete restriction site, right? And also some additional bases on either side, right? I mean, you don't have to have on either side, because once you fuse it here, on the other side you have other bases here. We have to ensure that there are some additional bases on this side. Why do we need that? It is to have the restriction enzyme interacting with the DNA efficiently, and then they will be able to make the cuts. Okay, why do we need linkers in the first place? Is if the fragment of interest or gene of interest does not have restriction sites that are compatible with that of the vector, then we can add the linkers that would have given upon restriction digestion, that would have given overhangs that would have been compatible with the products of the vector. So those are linkers. Then we also learned about adapters. Say in case if the gene of interest has a restriction site that is where we want to clone, then we cannot use the restriction endonuclease at that place. So there we can add adapters. Adapters are also double-stranded DNA, and they have overhangs which are something like products of restriction digestion. They resemble the products of restriction digestion. So these overhangs we will choose adapters, we'll synthesize adapters such a way that they will be compatible with the vector based on the context. There is one more important thing that we have to remember is they are made in a certain design. I'll put it this way. You can have blunt ends or other ends, either way. I mean, sticky ends on either side. Usually this one will be the adapter region or the overhang. I'm saying compatible overhang. So on the 5' side, we should be having phosphate, 3' OH, 5' phosphate, and 3' OH. But there is a difference, is that we will remove one of the phosphates from the sticky side of the adapter. So basically, this phosphate will not have been there. It would instead be OH, right? This is one design. The one that is described in this figure has one design where you have one phosphate and OH, and OH, 3 OH, right? Like is depicted also here. I drew it a little differently on the other orientation. So you can have 3' overhangs or 5' overhangs. The point here is that when we synthesize, we would have removed one of the 5' OH on the sticky side. Okay, and that is to prevent adapter ligation. We don't want adapter ligation. Because the goal or as the central idea of using adapters is to not use restriction endonuclease so that the fragment may not otherwise may cause internal breakage. If that is the scenario, in that case, adapter, if it is not present, it will if it is not dephosphorylated, it may ligate and it will form a linker, and then we have to use a restriction diation. So that's not a good solution. Therefore, we would have removed one of the phosphate groups to prevent adapter ligation. Okay, and we have also discussed. We were, I think, discussing terminal transferase. It is an enzyme. It is a DNA polymerase. And the difference is that it is template independent. And the important other part is template independent means whichever bases nucleotide substrates are available, it will just polymerize them. And the substrates would include DNTP, any of the DNTPs or all of the DNTPs, deoxynucleoside triphosphates, and they would also require a DNA molecule. DNA or RNA, any nucleic acid molecule. Basically, they need a 3' OH onto which, nucleic acid based 3' OH of ribose, and then they will add the bases. So if there is a DNA in this way, where this is 5', 3' OH, this is 5', 3' OH, this is not a good substrate because the overhang will impede the activity of a terminal transferase. But if we have something like this, where it is 5', 3' and 5', here is OH, and then terminal transferase or TT can add more and more bases. Even if they are blunt ends, right, 5', 3', 5' and 3', they will add nucleotides this way also. You should also remember that there is another 3' OH here, right? It can add the bases, polymerized bases on this side also. Okay, and here it can also base pair on this side. If there was a DNA molecule, which is something like this, right? This is 5', 3', 5', and 3'. The 3' OH are not that easily accessible. In this case, absolutely there will not be any polymerization. So it's worth thinking of it that way. So terminal transferase, we can use it to for homopolymer tailing and then ligation and then annealing and then transformation. So we can treat the vector by providing terminal transferase and GTP. So the 3' OH. On the 3' side, there would be multiple G's added. On the insert, we could have done it with multiple C's and then mix and anneal because G and C will base pair. And sorry. Sorry, I have some allergy. Okay, and then this can be transformed. And when they polymerize hundreds of bases. So there are enough hydrogen bonds, and upon a upon transformation, it could be repaired and the cloning is done, or recombinant DNA is formed. So the next ones are called, there are a few more, and then we'll get into, excuse me again. Yes. So after finishing TA cloning, we'll get into PCR. I hope you have done your homework on that. Sorry. So just give me a break. I'll clean up and come. Sorry about the interruption. Are you people there?
Yes, sir.
Okay, good. Thanks. Okay. TA cloning is something which is a little bit, it is a possibility and not many people use it these days, but it is still worth knowing it. And in some cases where I would also have usually an assignment, which is called as topo cloning. I hope you have already read topo cloning. Please try to have a look at that. So the point is, we will learn about PCR in a while, and we'll also learn PCR at a later time also. But right now, we would think to see what is TA cloning. TA cloning is where if we could develop only T as an overhang, and if for the product only A as an overhang, right, then the product is directly it will fit in here with the A this way and A on the other side. The sticky ends are matching and they are compatible. And if we add ligase, it will be sealed up and we have a recombinant plasmid. It's a simple approach. Point is, we should be able to generate these only A as an overhang or only T as an overhang. So what we should do to generate one of the common one, if it is TA cloning is used, is there are some restriction endonucleases which can generate these T overhangs, like in the case of HPH1. So this is the site of recognition and the site of cut is at a different place. This is not a traditional type 2 where the site of recognition and site of cut is in the same place, right? Usually they recognize, they bind at one place and kind of cut. There is no palindromic. It is not palindromic what we see of traditional restriction endonucleases. Anyway, the point is that they would result with T overhangs like this. And then if the vector has this restriction site, and these restriction sites should be on both sides. Say if there is a restriction site like this, and we should be able to have a restriction site reading from GGT, GAA, like in this orientation, and 1 in reading in this orientation. Only then, when this one cuts, it will leave a T overhang, and when this one cuts on the other side, you'll have another T. Only then we'll be able to generate a restriction endonuclease, the vector with T overhangs in this way. Otherwise, it's not possible to do that. Okay, and the same could be used. We have to search for restriction endonucleases that may leave a overhang that is compatible. There should be only one A, and that is compatible with the overhangs of the vector. One of the easiest or very common way is that during PCR, when we do PCR, if you are aware, we use tag polymerase. And tag polymerase usually leaves one 3' A overhang. And if we could have generated a overhang that is similar in nature, this is not 3' overhang. If you say 5', yeah, this is 3', sorry. Sorry, what I meant here is this is 3' T, isn't it? I'm talking about the vector here. If we are talking about the vector, it is leaving a 3' T, and this will be another 3' T. So the insert should be having a 5' A in that case. Okay, so we also have to match the overhangs properly, whether which one is 3' and which one is 5'. In this case, this is a 3' A. And the T, we should assume that this is the 5' T and 5' T. And then the product will fit perfectly into this place. Okay, so we will discuss about PCR in a while now, and I hope you understood the TA cloning concept. So this is one of the questions I have. What should we do in this situation? KPN1 is there, so restriction endonucleases that are available, BAMH1, and you know about it, GGA, TCC is for BAMH1, and for EcoR1, it is GAA, TTC. Sorry. So if we cut the DNA, we will get the DNA in this way, and on one side we will have BAMH1, the other side we will have EcoR1, and the vector could be cut with KPN1. How can we fit it in is the question. They're not matching, so we need to, KPN1 is this, and in that case we'll have to, there are multiple answers to this. One way is to use linkers. You can digest you can make them blunt ends through exonuclease. You can do blunt end cloning, right? That means you will make blunt ends on either side. You'll make blunt ends of the vector and do blunt end cloning. Instead, we can make blunt ends of the fragment and then add KPN1 linkers and perform that. We can also use adapters. If you didn't have, if you had a KPN1 site or any of those things, you could have used adapters to do the cloning. So there is not one answer for this. There can be multiple answers, and it depends on what is given to you. There are constraints or some limitations will be there. What is available to you and not. And there are constraints about the restriction sites, whether if they are present somewhere else in the fragment or vector. That is, those are other limitations. And based on that, you should be able to choose which of these is a best possible way for you. Okay, so think of multiple options all the time and find out the best answer. Okay, now we will try to go into PCR. We know about It polymerase chain reaction, right? So first thing we'll discuss about polymerase. All polymerases, DNA polymerase, and that's one of the reasons why we are discussing PCR here, is because we want to discuss about the enzymes. DNA polymerase and RNA polymerases. Both of them are important. The polymerization, the chemistry of polymerization is almost the same. A DTTP here, or DNTP, you can think of it, it has 3 phosphate groups. This is the base. This is the deoxyribose. If it is a DNTP, in the case of DNA polymerization, if it is RNA polymerization, it would have been ribose. And then we have 3 phosphates named as alpha, beta, and gamma phosphates. The 3' OH of the primer, this region we would call it as the primer, and the bottom one here would be called as the template. So the primer, the 3' OH here makes a nucleophilic attack with the alpha phosphate, and this phosphate, the rest, the 2, are left out as pyrophosphates, which will be again hydrolyzed to inorganic phosphates. And the base here is now incorporated into the DNA. And this reaction will keep on happening. So what are the substrates? For DNA sorry. For the DNA polymerase, we need DNTPs. And the DNA polymerases, as I described in the last class, are DNA dependent. DNA dependent DNA polymerases. I would also want to remind you, anywhere in the text, anywhere in your questions through chat, anywhere in the GCR or assignments answers, never write like this or like this. DNA is an abbreviation. Please write it properly, especially because we are biology students. Don't write like this. In part, at least, science is about maintaining the standards, right? Don't try to use shortcuts in science. If science is a formal thing, try to use it formally. Okay. DNA dependent DNA polymerase, RNA is also DNA dependent RNA polymerase. There are reverse transcriptase, if we will discuss them later maybe. Reverse transcriptase is RNA dependent DNA polymerase. Okay, it is opposite to the RNA polymerase. Okay, so we should remember, we will at points we will discuss about generation of in PCR, in part of the PCR, and also at later time radio labeling nucleic acids. We have a topic on labeling of nucleic acids. We should speak about end labeling, and we should speak about random labeling. In that case, it's important to know this chemistry. Okay. So DNA polymerases typically, in E. coli, if you see, for example, there are 3 DNA polymerases. Polymerase 2 is the one that is mostly used during replication. So there are, DNA polymerization. This is polymerase 3 is usually used for during replication. What you will see is polymerization rate, that is number of bases added per second, is very high here. And also, I hope you remember what is meant by processivity, number of bases added before the polymerase dissociates. And DNA polymerase is adding, for example, if this is the 3' OH, and how many bases it adds is at a time before the DNA polymerase falls off is processivity. And the sliding clamp, if you remember, which is important for this processivity. What we are discussing, what this table actually in general indicates, it's what happens in the cell. There is also another activity, 3' to 5' exonuclease activity. If there was a wrong base added, the DNA polymerase can move back and remove it. There it is moving from 3' to 5' direction. So that is the proofreading exonuclease activity. That is present in all the DNA polymerases 1, 2, and 3. As I was describing about Nick translation yesterday, DNA polymerase 1 also has an additional one, 5' to 3' exonuclease activity. So what happens in Nick translation, assume this is 3' side. Here is some other DNA. Using its 3', I will just talk about DNA polymerase 1 right now. It will keep adding more bases. And now there is no space here. Using its 5' to 3' exonuclease activity, it will start removing these bases. Using its polymerization activity, it will keep adding bases in this direction here. Okay, that's why DNA polymerase 1 is mostly used in repair. It can also remove even the RNA primers. But RNA primers during replication are mostly removed by primases. Is that right? Sorry, it is by RNase H. Primase synthesizes primers, and RNase H removes the RNA primers. Okay. So in PCR, everything, the one more direction we should always maintain, that polymerization always occurs from 5' to 3' direction. This is the 5', 3' OH, and then it will keep on going in this way. Now for PCR, what all we need, this is basically a crude way of discussing PCR. We will discuss this again at a later time. Right now, what I want you to understand is the general concept of PCR, how the polymerases are involved, and the most important one is about designing the primer. And that is one of the reasons why I'm discussing here, because it has to do with restriction diation and cloning. Okay, so here is the target DNA where we want the region to be cloned. And then we need to synthesize a primer. I will put it in this direction. I have separated the strands, 5' to 3', 5' to 3'. So I'm synthesizing some primers. Let's see how it is. I designed some primers like this. Let me write, this is 3' OH, and this will be 5' phosphate. 5' phosphate and 3' OH. If I have designed primers like that, and I add DNA polymerase, Taq DNA polymerase, what would happen? The DNA polymerization would occur in this direction, right? Because the polymerization is always from 5' to 3'. This will be polymerized on the other side. Then there is no convergence. Instead, I will design primers again. I'll keep the other ones there itself. This is 5', 3', 5, and 3. Now, if I have designed primers in such a way in this direction, right? This is 5', and this one is 3'. This is 5', and here is 3'. And we add DNA polymerase, then polymerization would have occurred in this direction. And for this, the upper strand, it would have occurred in this direction. So if we continue, let the extension happen, we would end up with 2 DNA molecules like this. So for PCR to start, one should ensure or design primers in such a way that the convergence would have happened. Upon polymerization, the direction of polymerization of both the strands should be converging, as shown also here. Okay, so the key or the first fundamental step in primer designing is to know whether you're synthesized primers so that they are converging while they're extending or not. Okay, and then that is the first step. And when we have a product, what if we can generate a product that would have had restriction sites here, some eco R1, say for example, so that cloning becomes easier. You can digest this with eco R1 and then put it up, ligate it into the vector. Yes. So for that, we can synthesize primers with where this is the region of homology, and they have added the restriction sites GAA TTC. You have to read from 5' to 3'. GAA TTC. The restriction endonuclease is present. You have added into the primer. You have added in addition to the primer. And I hope you know why do you need a sequence like this? Can anybody answer? GGCC. Why is that added? Anyone? Sorry.
So that the polymerase could come and bind, interact with that particular strand for stability.
What polymerase?
The Taq polymerase, sir.
Why? Why would you need this sequence?
To increase the stability of the interaction.
Interaction for what? I can try. I think you are on the path. I just wanted to try to squeeze the answer from you. Okay. If the product has already formed and you did not have this, what would the sequence be? It would be this much only. This part would not have been there. Right? The same is on the other case. On the other case, they have added a Hint3 restriction site and they put this one also. So if that was not put, the end product would not have had this region. Now for the restriction type, you will add eco R1 and Hint3. If you remember in the last class and also in this class, I discussed about the restriction endonucleases. This is the restriction site, the TTC. They would require some additional bases to be on either side of the DNA to make proper interactions and cut effectively. If we have not added this region, okay, then the end product will not have these regions that I struck off. This is the restriction site, AAG CTT. And this is the restriction site, GAA TTC. The restriction endonuclease may bind but not be very efficient because on the other side there are no bases. Okay, I hope you're able to see what I'm trying to imply. Right? The reason, if we have included these CCGG here, the ultimate products of PCR will also have this. Therefore, when the restriction endonuclease binds, when we add eco R1, they can make proper interactions with the with the DNA, target DNA, and cut them efficiently. That is why we add these things. Okay. The same is the case with when we discussed adapters. I think I have given you in greater discussion we had or linkers. Sorry. I think when we are discussing about linkers, this is the region for BAMH1. We added 2 more bases here. Why do we need those additional bases is the question, right? To increase the stability of interaction and restriction. Some restriction endonucleases or most restriction endonucleases require these additional bases on either side. That is the reason we need to have. So yes, I hope that is clear. If there is a question, we can always discuss that. So primer, if you look into it, you have 3 different parts. First one is the region of homology. This is only for cloning primers. Cloning primers. There are other primers just amplifying. That means they do not have any other purpose, just PCR, and you will just run the analysis. It is an analytical one. Amplifying region primers are only require the regions of homology. Okay. Cloning primers require additional, this is the restriction site that we are going to add. And then you have these additional bases. I used to, in my lab, when I was working somewhere, we used to call it as buffer sequence or something like that. Each lab may have their own names and so on. Okay. Cloning primers have 3 regions. Region of homology, and the purpose of this homology is to give specificity to which region should be cloned. We would have used the primers that are specific to the region of interest, right? Only then we will be able to get the amplify the region of our interest or gene of our interest. The second part of it is the restriction site, so that we will be able to cut and make the compatible ends so that we can clone it. And then we have these additional bases. This is to increase the, during restriction, these bases are useful for interactions of restriction endonuclease with the product and cutting of the DNA. Okay. So if you understood this, I would want you to design primers for this. I want you to design cloning primers. Okay. And you can choose any of these restriction endonucleases. And this is also important for directional cloning. We will get to these again as well, just for, I think it's good and easy to talk about a topic twice. In many cases, I discussed, in some cases, it doesn't matter in which direction you clone. In some cases, I gave you an example or a problem. I want that this cloning should be only in one direction. This is promoter, for example, and here would have been the start, and this would have been the stop codon. So only when expression, when transcription happens, mRNA is produced, the correct one, which will then give rise to a protein that corresponds to this particular gene. So if you want to do directional cloning, you have to ensure that on only the open reading frame is downstream of the promoter, and that the start codon is upstream of the stop codon in this orientation, basically. If you want to do that, and as you if you want to ensure that this is directionality is maintained, then we can use we can try to have, say, for example, ATG is the start codon, right? 1, 2, second codon, or third codon, 4, 5, and 6. TGA is the stop codon. The other stop codons are TAA, TAG, and the other one is TGA. Okay. So I have made this bold to indicate that just use that as a region of homology, just 3 bases. Typically, the primers are bigger. We will discuss that later, but for now, just try to get the concept. I want you to first step is make this double-stranded. In your books, you can make this a double-stranded one. Then you can denature them. Basically, what I meant is write the second strand, the complementary strand, in this line here. Okay. So I will go about it with you. So probably I am hoping that you are also doing with me, because that will give you a lot of practice. My region that I need to clone is from here to here, which is from here to here, right? And I'm looking for primers to clone for cloning a fragment. If you remember what I discussed about what I want in a cloning primer, we will need region of homology, the restriction site, and I want to have, we should have additional bases as well, 3 or so. So because I want it in directional cloning, I can choose based on what vector is available. Say, for example, the vector that is available is this promoter is here, and it has a multiple cloning site. We will discuss all these over again. Don't worry about it. So I'll put here BGL2, and for simplicity, we will say KPN1, right? So I need to get a fragment such a way that the open reading frame is here, ATG is here, TGA stop codon is on this side. And I should get a product that has the restriction site for B or BGL2 here, and KPN1 on this side. And then when I cut it, say for example, it is going to be cut like this, and this one is going to be cut like this. Okay. So this B will fit. If I cut the vector with BGL2 and KPN1, I will get B overhangs on this side, and I will get K overhangs on this side, KPN1 overhangs. And if the vector, if the gene of interest is cut with BGL2 and KPN1, it will be a perfect fit into this one in the correct orientation. So we need to now design primers. So I will try to make mistakes. Let's see how it goes. I'll make a primer this way, and this is the region of restriction site and some additional bases. Region of homology is this way, CAT. If I do like this, the problem is this is 3' and this is 5'. If polymerization occurs, it will happen in this way. It's not right. That is wrong. So we cannot use that. Right? I hope you understood that. So instead, what we should do is choose this as region of homology, GTA. As I said, region of homology is usually 20 to 25 bases, or sometimes it depends on the situation, but typically they are somewhere in this range, nucleotides. Restriction sites is typically 6 bases, and additional ones could be 2 to 4 or 5 bases. It depends on what type of restriction site, the requirements of the restriction site. So I am choosing, for sake of understanding, I'm choosing the, in this for demonstration purpose, I'm trying to take only the region of homology, just 3 bases, because it becomes difficult to describe that. And then I need to have a region of the BGL2 site, as I described here, right? BGL2, you have to be careful, everybody, all the time. about in what orientation we are writing the sequence. So this is the restriction site I wrote, AGA TCT, like in here. As I said, AGA TCT and TCT AGA are two different restriction sites, because reading always happens from 5' to 3', then this sequence is AGA TCT. It is never, it is not the same as TCT AGA. Okay. And because BGL2 we have added, and we need to have some additional bases here. Say we can take CG or CG, CGG. We'll do the same for the other primer as well. We will take the primer here. So I'll take site. The sequence would be TCA, right? That is the region of homology. And then we need to have a different restriction site. That would be this one. And you have to remember this is 3'. This is the 5' side. And you have to write it in this way: G, G, T, A, C, C. Then it is KPN1. If by mistake we have written it GGA this in the other ways, it is wrong. It is not KPN1 site, it will be some other site. And then we will need some more additional bases, C, G, G we will put, right? I hope you understood the three parts of the cloning primers: region of homology, region of restriction site, and some additional bases, right? On the same way in the other primer as well: region of homology, region of restriction site or restriction site, and then additional bases. The other simplest way, other ways of doing this is something like this. I will just draw it and I'll stop there. So I want to clone in this way, B and K. Then for designing purposes, I would have written the restriction site here. The gene of interest in this way: AGA, ACT. Then what would I need? I would need BGL. I need BGL2 sequence. That is AGA, TCT. And then additional sequences, any of them, GCC. And here I would need the restriction site. Restriction site is KPN1, which would be GGT ACA. Is that right? No, ACC. When I'm writing the bottom strand, I'm trying not to speak. You understand? Not to speak or read the alphabets as it is, because I should be typically writing it from this side to this side, but read this side to this side. So if I was reading it as or representing the KPN1 site, I'm saying CCT, CCA, TGG, then it is wrong. That's why I'm not speaking it, right? If reading should have been done only from 5' to 3' side. And then we will need some additional sequences like this. Now it is easy for me to design a primer. I'll see if I can pick. Sorry. So if I get, upon PCR, if I get a fragment like this, then it can be digested and cloned into this one. So I write the product. Theoretically, what product would I want? And from there, I can pick this as one of the primers. That is this one. And I can pick this one as the other primer. That is this one. Okay. And then Those are my cloning primers. But anytime you are representing, you have to write it in 5' to 3' direction. Even if you are ordering, it should be in 5' to 3' orientation only. Now, writing the bottom strand is the problem. You call this as forward primer. The other one could be called as reverse primer. It depends on the labs and so on. Now I have to be careful and to write it TAT, GGT, ACC, and TCA. Right? Because both are being written in 5' to 3' 3' orientation. If you want to send it to a synthesizer, a private person, it doesn't matter to them what is reverse and what is. He will only synthesize what you give. And typically, every nucleotide is represented always from 5' to 3'. And when you're ordering, you order them as single nucleotides. You don't order them as double-stranded DNA and all those things. That's your headache. Synthesis is the only part they would do. Right? I hope you understood some concepts of PCR and primer designing. This is one of the questions that will come. A sequence will be given, a scenario will be given like this, and then you are supposed to design the primers. And I hope we have covered this. We will redo it if required. And this is your job to find out the precautions one should take. And you should know what are primer dimers. Okay, please try to know them, how to avoid them, what is the risk of they being in the reaction itself. Okay, if you have any questions, I'll take. Otherwise, you can leave if you have other classes or get ready for the next class. If you don't have, you can leave. If you have any questions, you can just ask.
Sir, I have a few doubts.
Yes, please. The
So with
others, you
respect
can
to-
stay or leave. It's up to you, okay? No issues. Parvati, go ahead.
Sir, with respect to terminal transferase, you mentioned that there is, like, could you please take the slides, sir?
I'm trying to get there. What did you say? Terminal transferase?
Terminal transferase, yes, sir.
Here? Yeah. Tell me.
So, in the fourth possibility, like having two
This one?
overhangs, yes, sir.
This one, yes, okay.
You mentioned that it is not possible for the terminal transferase to go work there, but then still we have, because there is 5' overhang. If there is a 3'
Yes.
overhang, then it is
Even
possible.
primary overhang, no problems. Yes, this is to
Okay.
imply that it cannot, if there is this extra, then it is a headache. It's not about whether it is overhangs on both sides or not. Yes, certainly, if it were 3' overhangs, that is a best substrate for it to polymerize there. Okay, in this case,
Thank you, sir.
polymerization will happen, no issues at all. Yes?
Yes, sir. Thank you, sir.
Yes. Next one.
It's with regards to the adapters, sir. You mentioned that there can be blunt end adapters also. So, what?
No, blunt end adapters, what I meant there is on one side, blunt ends. On one side, or say, for example, I have a DNA molecule like this. Okay, just making up. Okay, this is an This is the gene of interest. It has overhangs. How can you bind the adapters to it? How will you design adapters to it? There are two ways you can do. One is, say, this is an adapter. One is that way. The other one is to make an adapter something with this like this. If you designed adapters like this with blunt ends on the other side, you should have filled this gap or did something to make the vector also blunt end. Yes?
Okay, sir.
If not, if you made compatible ends here, compatible to the overhangs on the fragment, you can directly ligate it here.
Okay, sir.
That's what I meant there.
Yes, sir. Now it's clear, sir. Thank you, sir.
Okay, chill. Any others questions? Anybody? Do you have, or that's enough? That's it.
Sir, I have a
Parvati, do you have any other
request,
questions?
not with respect to the class, sir, but then I have a request with regards to the assignment.
Okay, what is it? I'll stop the
Like,
record. Okay, tell me.
yes, sir.